Our last speaker for this session is Dr. Daphna Rothschild from Stanford University. Daphna, the floor is yours.
Hi, thank you. I am very happy to be here. Let me share my screen. So I want to thank the organizer for giving me this wonderful opportunity to be here today and present to you my work. I hope you can hear me, right? Can you just, is it okay? Okay, great. So I will start. And in my work, I study ribosomes. And usually when we think about ribosomes, we think that these are very abundant machineries in the cell that just passively translate which mRNA is found there. But I hypothesize that there are different types of ribosomes. One type of ribosome that's found within the brain synapses. These are long-lived cells and we don't want to accumulate misfolded proteins. And maybe other types of ribosomes that's found within the gut epithelial cells. These are cells that only live for a couple of days before they're shed off into the gut, and they're exposed to harsh conditions and they have high protein secretion. And I hypothesize that there could be trade-offs. For example, the translation speed of the ribosome and accuracy of protein creation. And I focus on ribosomal rRNA genes. You can see here in the cartoon, they're found in the core of the ribosome. And what's unique about the ribosomal rRNA genes is that while most genes are found in the genome in a single copy, ribosomal rRNA genes, they're the most repetitive genes in the human genome. They're found in hundreds of copies across multiple chromosomes. And you can zoom in to one of these chromosomes, you can see the tandem repeats one after the other. And already in the 70s, it was observed that there are sequence variations between the different copies, but because of technological limitations, we didn't know their identity. But there were two papers that did study the variations across the population. The first one being in 2018 by Parks et al., studying the 1000 Genome Projects and reporting hundreds of variants. And then 4 years later, Fan et al. Reanalyzed this paper and found major limitations, wherein particularly the 1000 Genome Project carried short-read sequencing. And short-read sequencing do not sequence highly GC-rich regions. And from their paper, what you can see is they're plotting the rDNA locus, and you can see there's inverse correlation between GC content and sequencing read depth. And there are regions that are just left completely unsequenced, and we don't know which variants are found there. And these include fascinating regions called expansion segment regions. So what are these expansion segment regions? These are fascinating regions. You can see here the bacterial cartoon and the structure, and you can see in gray the ribosomal rRNA, and in blue you can see ribosomal proteins. And across evolution, what you can see is that the ribosome have massively expanded, specifically in the ribosomal rRNA, and you can see in red these expansion segment regions. And what's unique about these regions is that they're found on the outer surface of the ribosome, and that makes them capable to potentially interact with proteins and transcripts to facilitate translation. And because they're found on the outer surface, they're flexible, and we actually don't know their structure, so they're still unresolved by cryo-EM. But while we don't know their structure, we do know that the expansion segments have functions, because if you create a truncation in one of these long expansion segments, you get misincorporation of amino acids, and they were also shown to bind transcripts. So in my work, I ask which variants are found in the rDNA, especially in the expansion segment regions, which are masked because they are highly GC rich, and which of the copies that carry variants is active, whether the variations form translating ribosomes, and whether these variants are functional. And this is a harder question that we're only beginning to understand. And I will share the results that were recently published. So starting with the first question, which variants are found in the rDNA region? I reanalyzed the 1000 Genome Projects, and I mentioned it had short read, but now for a subset of individuals, it also have long read hi-fi sequencing data. And to analyze the variants in the hi-fi sequencing, I developed a novel algorithm for variant discovery between these full-length paralog genes. And when I analyze the long read data, I find in striking contrast to previous findings that found mainly single nucleotide variants, I find that most of the variants are actually insertion deletion variants. And to see that this is not an artifact of hi-fi, I replicated this result in another independent dataset called the Genome in a Bottle that has both hi-fi as well as Oxford Nanopore. And I see that both Nanopore and hi-fi agree that insertion deletion variants are the main variants of the rDNA locus. And this is just an example of one of these expansion segments. You can see that these are extremely GC rich, even without reading the sequence. You can appreciate homopolymers of Gs and Cs. And I was able to show that they, of course, carry now indels, but we also can actually find these in the short read data. Just they're so depleted that we don't find many reads in these regions at all. So I asked whether these variants are active, whether the copies in the genome form translating ribosomes with these variations. And to do that, I collaborated with my lab member, Theo Susanto, and we developed RiboRT to sequence the ribosomal RNA from the translating ribosomes, where specifically in the naturating conditions, we were able to completely reverse transcribe from the large subunit and the small subunit, the 18S and the 28S rRNA. And the approach that we selected was to take a human embryonic stem cell line and to sequence both the translating ribosomes as well as sequence the genome to compare which copies are the ones that produce the translating ribosomes. And we used the variant algorithm that I described earlier to find the variations. So before showing you the results, I'll just explain what you're going to see in this plot. You're going to see variant frequencies in their frequency in the translating ribosome on the x-axis and their frequency in the genome in the copies on the y-axis. And what you can see is that there are many dots, there are many variants, and most of these variants are found lowly expressed. But there's also a subset of variants that are highly expressed. And you can see a correlation. Specifically, variants that are found across many copies in the genome are the ones also highly expressed in the ribosomes. And this serves as evidence that these are coming from different loci. So what are these variants? I'm going to show you like one example. Here you can see that the reference allele in this particular position is a G, but actually we also see an A, that's a substitution, but we see both of them in high frequency. And let's look at another example. Here you can see a GGC, GGC, that's a tandem repeat. And this can be deleted or have one copy of GGC or even different number of these repeat. Now looking at the other high abundant variants, you can appreciate that many of these are insertion deletion variants. But what's most interesting about these is that these are not just found randomly positioned on the ribosome, these are found on the expansion segment regions. In 4 of these expansion segment regions, again, these regions on the outer surface of the ribosome that we think are interesting because they have the capability to potentially interact with different proteins or transcripts. And we continued to follow on these high abundant variants, and we collaborated with the Broad Institute, with the group of Xiao Wang and her student Xing Sui, and we developed SwitchSeq, which is a method to sequence these ribosomes with the variations. And we can see these variations in single ribosomes within single cells. And this method now allows us to follow these variants spatially. So we asked, are these variants functional? And do these variants affect the ribosome? And as a first functional readout, we asked whether these variants change the structure of the ribosome. And to do that, we again sequenced the ribosomes, but this time we added DMS, where DMS modifies the RNA, but only where it's accessible. So we can compare the accessibility of different types of ribosomes. And I'm going to show you now differences in the structures in 2 very abundant subtypes, if you could call, ribosomes. Here showing you like a green type of ribosome and pink ribosome, and they vary in their nucleotide sequence. So zooming into the DMS results, what you can see is that around the region where you have the sequence variation, there is a change in the stem loop predicted structure. As well, if you just look at the coloring where the DMS was bound, you can see that the pink type of ribosome has more DMS bound compared to the green type of ribosome. And we hypothesize that this could be because there could be some ribosome-associated protein that can bind the green type of ribosome, but not the pink type of ribosome. And we ask, if these are different subtypes, where are these located in the genome? And to do that, we analyzed the telomere to telomere. They released a complete human genome in 2022, including positioning of the rDNA units. And we find there that the ribosomes are chromosome specific, where, for example, I color-coded chromosome 13 in the color purple, and you can see that this particular chromosome carries 76 copies of the rDNA, and 73 out of which contain the same composition of variants. And so we created this catalog of human expressed ribosome variations, and we asked, can we now use this atlas to see whether there are changes in expression of ribosomes across health and disease? And I'm going to show you here with the very short time results. When we compared the expression of ribosomes across different tissues, we analyzed the GTEx dataset. And you can see that this green type of ribosome is found higher expressed in the cerebellum compared to the gastroesophageal junction. And this held true, looking most pronouncedly the difference between digestive and brain tissues. And we also looked at another cancer data set. There we compared different cancer types, and there we see that there are variants which are usually found lowly expressed in control biopsies, but become elevated in the cancer biopsies. And here I'm showing you an example of colon adenocarcinoma, but this holds true for different cancer types and different variants. So to summarize my work, I showed that insertion deletion variants are the main variations in the rDNA loci. These include novel haplotypes, which we term as novel subtypes. As we show that they have differences in their structure, we can visualize them, and they're localized on different chromosomes. And we created an atlas of human embryonic variants, but we can now also use this atlas to quantify ribosome variations across different conditions in any sequencing dataset. And this is only the beginning of our understanding of what could be a novel layer of translation regulation by ribosomes with variations, because what we want to understand is whether these different ribosomes translate unique mRNA pools, where these are located within the cell, whether they actually affect cancer, and whether they are important in development. So with that, I will thank my advisors, Maria Barna and Jonathan Pritchard, my collaborator, Theo Susanto, that I mentioned in our collaborated from the Broad Institute, the Barna Lab and Pritchard Lab members, and funding. And I will be happy to take any question. Thank you.
Wonderful talk, Daphna. And we now have some time for questions.
I have a question. Hi, Daphna. Thank you for a very cool talk. I was wondering if you have tried, or if you can link the different ribosome variants to different tRNAs or different mRNA preferences along those lines.
So first of all, I didn't do it, and that's actually really interesting because you're saying there are different tRNAs, but they also carry so much variations, modifications, and also for ribosomes, you could go and ask similar questions, whether modifications on the ribosomes are affected by the variants. The simple answer is I just didn't check it. It's very interesting.
Cool.
I think it was a great talk. Thank you very much. And I wonder if there is any link between rRNA variation to diseases in wide population of patients. So is there any correlation between specific? That's the first question. The second one is in many neurological diseases, there is a problem in protein homeostasis or translation. So any link between these two, protein translation in disease?
Okay, so I had only very limited amount of time to show. I showed just a glimpse from cancer. There, deregulation of translation is pretty much a hallmark of disease, and we can see that variants associate with different cancer types. And I didn't look at neurological disorders, but I am looking now. Actually, you asked about other diseases. I am actually now analyzing on the UK Biobank, and it does carry a few diseases. It's a healthy cohort in general, but we are actually powered now to see that we see even at the level of germline mutations that they associate with asthma and other diseases. Oh, but there was a second question. I'm sorry.
Answer both. It's great talk. Thank you.
Thank you.
I see that Abigail Stoker has a question. You may speak.
Hi, thank you. Very interesting. I actually wanted to zoom out a bit from our interest in humans and ask if you see this in other organisms across the tree of life.
Oh, thank you. This is a really cool question. So I'm very interested in bacteria. I did my PhD on gut microbiome. There, it was actually shown. So actually, in all organisms, you have multiple copies of the rRNA. And, in bacteria, that's how we classify the identity of different species by 16S ribosomal rRNA variations. We don't normally think of these to be functional. We just use them to classify different bacteria. But actually, in bacteria, in a specific Vibrio pathogen, it was shown that there are multiple copies of the ribosomal rRNA and that they carry sequence variations that are functional. And they showed that if you grow this bacteria in different conditions, they express different types of ribosomes. And it was actually shown that if you modify, you create a strain that of this Vibrio that doesn't carry a particular copy, composition of variants, that changes the virulence of this pathogen. They give it to a mouse, and that changed whether that mouse survived or not. So it does have function, at least in bacteria, and that's kind of what we know. We don't know much about other organisms.
Okay, we have one last question that I see that is written here in the chat from Mike Feinzenberg. Any idea what kind of functional differences might occur between these ribosome subtypes?
So I do hypothesize that the differences in the what I showed is that there are again, I had very limited time. There are many changes that are happening in the structure of the ribosomes, particularly where we see indoles. Specifically, there are tandem repeats, GGC, GGCs. These GGCs were suggested to form quadruplex, like they have special function, special structures. So I hypothesize that these changes in structure change which proteins that potentially can recruit mRNAs to the ribosomes. Or, alternatively, I hypothesize that that maybe, again, there could be trade-off like speed of translation. That is what changes between these different subtypes, the speed of translation, and that could, affect different needs in the cell.
Thank you, Daphna, and thanks to all the presenters in this session.